Okay, thank you. Bye. Bye. Hi, good evening, Anant. Hi, good evening, everyone. I know it's the time now. Let's wait for 5 more minutes and see if people are joining us. If not, I'll start within 5 minutes. We'll wait for 2 more minutes and then I'll start. Yeah, I've waited for 5 minutes. Now let's start. Hello, everyone. Welcome. Uh, Good evening. I'm Gitanjali Singh, your TA for Medicinal Chemistry. I'm a PMR fellow from Department of Chemistry at IIT Kanpur. Hi. So I'll be taking this problem solving session every Monday for the next 12 weeks. Every Monday from 6 p.m. to 8 p.m. You all have been provided with the link that you used to join, right? So this link would be valid for the coming 12 weeks and it would be the same link. And during this sessions, I would like to summarize whatever we have been taught throughout the week. I'd like to solve the last week assignment and give you some hints for the upcoming assignments. And yeah, I'd be glad to join any kind of queries that you have regarding the topics that were discussed that week. In the upcoming weeks, as we move forward with the subject, there'll be certain topics that we can cover gradually. Sometimes you might be curious and it might sprout out some topics. Please wait. I'll try to explain it to you, but sometimes some topics need in-depth discussion. Would wait for the topic to be discussed, um, or else I can just give you a small preview. Nevertheless, I'd be more than happy to discuss any queries that you guys have. This session will be recorded and again shared with you. I'd prefer if you guys could join live. That way, we all can have a very helpful discussion regarding the topics. And if you have any queries that come you in the sport of movement or anything like that, I have muted everyone. I would like you guys to be muted because this is going to be recorded, and that would mean smooth recording of the session. But if you have any question, anything, please post it in the chat box. I'll make sure to check it regularly. If some topics, I would like to clarify everything then and there. But if there are some things that would require in-depth discussion, I'd like to take that at the end of the session. But please put there. You can give your reaction and unmute yourself to ask a question as well. So yeah, I have modeled this session in a way that I have written all those questions, and all those questions are, in a way that we can, gradually how the topic goes on. So we'll discuss discuss the topic in form of questions. I'll tell you why the questions are right, why it's wrong, and we can have a better discussion of the topic. So let's start. In this week, we have covered Introduction to Medicinal Chemistry, Part 1 and Part 2, Intermolecular Binding Forces, Protein Structure and Functions, Tutorial 1, which includes basic concepts of acidity, basicity, and some related concepts that you might require for your further lessons, and Tutorial 2, that includes basic concepts of thermodynamics and kinetics. These are all in basic level, and you have been given the assignment called assignment 0. It was not supposed to be graded, but I hope you guys had a look through the assignment, and we can discuss that assignment as well. Now, the first question: what is the definition of medicine? A chemical substance used for recreational purpose only. A drug or preparation used to diagnose, treat, prevent, or alleviate diseases. C. A compound exclusively derived from natural sources. D. A substance that only cures bacterial infections. So, have a look through the question and please try to post the answer, whatever you think is in the chat box. I'll give you 1 minute and then we can see. Yeah, okay, I've got all the answers, and all the answers are right. It's B, a drug or preparation used to diagnose, treat, prevent, and alleviate diseases. Basically, medicine is any kind of drug, and by drug, I mean any kind of compound that has, we'll talk about toxicity later, but a compound that we use to diagnose, like to know what the kind of disease a patient might be suffering from, and try to treat the medicines should at least treat, if not cure, treat the person in such a way that it reduces the symptoms, makes the person more comfortable, and you know if not cure or treat, at least prevent. We have certain medicines that are more preventative, and they work for deficiency or something like that, so to elevate disease. So that's like a very basic concept of what medicine is. So that's the correct answer. Well done, everyone. Let's move on to the next question. Which of the following is a common method used in drug discovery? A. Random testing of chemical compounds without any target. B. Target identification followed by high throughput screening of compounds. C. Manufacturing drugs without any prior research. D. Using drugs from traditional medicines without any modification. Take a minute, guys. Have a look through it and give me your answer in the chat box. If you have any query regarding the question or why this is not correct, please ask me, and I'll be more than happy to, answer that. So I think we've got the answer. It's again B, target identification followed by high throughput screening of compounds. Now let's see why the first one is not correct. A says random testing of chemical compounds without any target. Well, if you have lots of things that you know are active, so they say, okay, this is a medicine that works for say cold, and this is a medicine that works, that lowers down their fever. So there are so many kind of medicine. You can just keep on testing, like, what if this medicine works on this one, it should work on this one, it should work on this one. You don't know what the target is, you're just randomly testing. We cannot do that because, one, that's not efficient. Number two, it does not serve any purpose other than waste your time. The second one is target identification followed by high throughput screening of compounds. Now, this is how we do it nowadays, because what we want is, we have all so many data available to us. How the drug discovery works nowadays is, we identify the target, like basically the protein or the enzyme that specifically is responsible for the disease, a mutation of certain kind in the enzyme, or anything that causes that disease. So what we do is, once we find the target, we design we design a compound based on the target, like, something that will have more chance of having some kind of activity to basically either stop the action of the enzyme, or maybe increase, if the enzyme is not working, maybe help it work in a way that, that disease, it no longer causes any disease. High throughput screening is basically having a large library of compounds and going through it to search the best lead, and the lead is the compound that has the best activity for the target that we want to work against. Now, manufacturing drugs without any prior research, that itself is absurd. First of all, there is no market value for a drug that has no research, no R&D, and it serves no purpose. And the uh, last one, using traditional medicine without any modification. Now, the thing is, traditional medicines, most of the time what happens is, we use some things that are based on our ancient knowledge of how things work. Like, we know that turmeric is antioxidant, so sometimes we use it for antiseptic purpose, and it is also antiseptic, and neem and things like that, but we don't know exactly how it works. But it has been passed on to us from generation to generation, so that we know, we more or less have some kind of idea of how that thing works, but not exactly. So we don't know what its potential is, and if it's causing some kind of toxicity or not. So it's better to, have some kind of modification to increase its efficiency and decrease the toxicity, and basically, make that medicine more efficient. That's a correct answer. Now let's move on to the third question. Which of the following correctly represents the stages of drug discovery? You guys have a look at it, and then I'll explain the stages of drug discovery. I can explain every stage, and then you can decide which one should be first and which one should be later. So, clinical trials, target identification, preclinical testing, and lead optimization. So, preclinical testing, and I'll let you guys. So I see you guys have done that. So again, the correct answer is B, target identification followed by discovery, then we go for preclinical testing, and then clinical trials. Target identification, as I said before, we first try to find the protein or the enzyme or anything that we can target. It's easier to, basically model a compound after identifying a target. It can be receptor, um, maybe any kind of lipid or carbohydrates, anything that you want that can be easily targeted, more or less easily targeted. Once we find the target, we go through sometimes we design the molecule, and if we have some leads on that, like if we have some data based on earlier research, we go through lead discovery. Now, what is lead? Lead is basically a compound that has like the best activity, more or less a scaffold that has a better activity against the decided target. Now, after that, we go for preclinical testing. Now, preclinical testing is like formulation of a compound, and to check, the synthesis, the formulation, to check the stability, toxicity, a very basic stage, and then clinical trials. Once everything is approved, I'm sorry. Once everything is approved, the animal model passes, then you go for a clinical trial. In clinical trial, there are certain stages. You might have heard of phase 1, phase 2, phase 3. They go on with, once the animal model is approved, obviously they go for testing in humans. Now, humans, first they check that with healthy people, you know um, around, maybe a group of volunteers that actually are paid, and that's basically done for. Yeah, I think I'll follow that in the next question. And this much is enough for this one, I guess. Yeah. So, guys, this is the fourth question. This is the match. You have to match group one, group two, group three. This is the one that we got, that you might have got in your weekly lessons as well. Please try to do this, and then we can discuss more on this question. Please put your answers in the chat box so that I can get the idea whether you guys have any problem with it. If anyone has any problem with it, please let me know. I can go much more details. If not, if everyone understands this, we'll just try to move ahead with a very basic idea of what it is. I am audible to you guys, right? Like, is the voice clear? Okay, I would take that as a yes. I'm still waiting for any answer in the group, in the chat box. Okay, yeah, you're right. Yeah, you guys are right. So it is A. So in phase one, what we do is we have a group of volunteers from around 50 to 100, or maybe even less sometimes, a group of healthy individual who basically volunteer for this kind of testing. Now, these guys are given this medicine which has been formulated, and it has passed the animal testing and everything, in vitro, in vivo, things like that. So these are done basically to check the pharmacokinetics, you know and everything, how the body is processing through it in healthy individuals. Now, once that gets it's passed through, we enter to the second phase. Now, second phase is phase testing is basically a group of a diseased individual. Now, we know that these guys are suffering from the particular disease for which we have designed the medicine. Now, these guys are given the medicine And this time it's done to see the dosage and everything. And the third one is in which we increase the size of the sample. Like, they go on from 100 diseased individuals to like 1,000, 1,000 diseased people. And here's when they see, the adverse effect or things like that before putting it into market. So those are the three phases of, testing. Now let's move on to the fifth question. What are the targets in drug discovery? So we have been talking about targets. What actually are these targets? Like, how do we actually find what should be our target, and what can we target? Human body is such a, organisms have so many kind of things. So what are we targeting when we say target in drug discovery? One, A, enzymes and receptors. B, carrier proteins and structural proteins. C, nucleic acid. D, lipids. E, carbohydrates. F, all the above. Hello, I see someone has raised query. Just a minute, let me see if people are wanting to come in. I'm sorry, guys. Yeah, I can see almost everyone has selected G, that is enzyme and receptors, carrier proteins, and structural proteins, nucleic acid. Why not lipids and carbohydrates? Okay, the correct answer is F, all of the above. Now, I can see why there's a confusion. Actually, we can target lipids and carbohydrates as well. So, it seems like you guys know well about enzymes and receptors, carrier proteins and structural proteins. So, and nucleic acids as well. But there are few drugs we can target actually lipids as a target as well as a carbohydrate. Now let me explain why how we can target lipids. You see, there are certain. Let's take an example of antibiotics. So our cell wall is made up of lipids as well, right? So in order for an antibiotic to work, the cell wall can also be a target, right? It can directly go and rupture the cell wall and cell membrane. We have that lipid membrane as well. Targeting a lipid, though not very specific, can also kill the bacteria. So in that sense, lipids can also be a target. And now we have not been very, I'm sorry, wait. And now lipids can also be targets as well as carbohydrates. Now carbohydrates are present in cell membrane as form. So you know how different kind of cell recognize each other. So carbohydrates are present in cell membrane, they're embedded there, and they are used for cell recognition. That's how a sperm identifies an ova. And if we target, and some cells, especially cancer cells, they have different kind of, not in all kind of cancer cells, but there are few cancerous cells in which they have different composition of carbohydrates that are used for cell recognition. And once we're able to target them, we get a very good anti-cancer drug. So actually all of these can be used as targets. Do you have any question? Please post it there, or you can unmute yourself and ask the question. Should I move ahead, or if you I'll take that as a yes and I'll move forward. Okay, another question. What is a lead compound in drug discovery? Yes, this one is good. So, A compound that is ready for clinical trials. B, a compound that shows promising biological activity and serves as a starting point for drug development. C, a compound that has passed all safety checks. And D, a compound that is used as a placebo in drug trials. Yes, guys, the correct answer is option B, a compound that shows promising biological activity and serve as the starting point for the drug development. Now, a compound that is ready for clinical trials is more or less a formulated medicine that has passed all animal testing, in vitro, in vivo, things like that. So it's more or less a medicine that is waiting for approval. C, a compound that has passed all safety check, then it's already more or less ready for market medicine. D, a compound that is used as a placebo in drug trials. Now, placebo is when a group is tested, a section of a group is given placebo. That's not actually a medicine. So let me explain how this trial works. So when sometimes what happens is when what you're taking a medicine that is supposed to have some kind of some kind of activity, your brain sometimes tricks you into believing that it works. So placebo, that's called placebo effect. So what they do is they give some kind of sugar or some kind of very harmless non-toxic thing that does not have any effect, neither good nor bad. So a section of people are giving this placebo and a section of people are actually given this medicine, and then they check the medicine, then they check the efficacy of the medicine. That's how it works. So yes, Vishal, you can get this PPT. I'll post this later. This is all recorded. You can get the session, the recorded session, as well as the PPT from the drive. So once the class is over, you can get everything. So let's move on to the next question. So, I'm sorry. Huh? Just a minute, guys. I don't know why it's happening like that. Oh. What does pharmacodynamics primarily focus on? A, how the body processes the drug. B, the therapeutic and toxic effect of a drug on the body. C, the movement of drugs through the bloodstream. And D, the rate at which the drug is eliminated from the body. Take a minute, guys, and answer this in the chat box. So that's a correct answer, Tanmay and Shweta. And the answer is B, the therapeutic and toxic effects of a drug on the body. So, you know, when we talk about medicine, there are two parameters, two main things that we discuss: pharmacodynamics and pharmacokinetics. Now, pharmacokinetics is once we ingest the medicine, how our body starts working on the medicine. That's called pharmacokinetics. And pharmacodynamics is what the drug does to our body. See, so that's the correct answer, the therapeutic and toxic effect of a drug on our body. Let's move on to the next question. What is the primary goal of pharmacokinetics and drug therapy? A, to study the mechanism of drug action at the cellular level. B, to determine the most effective drug combinations. C. To understand how the body absorbs, distributes, metabolizes, and eliminates drugs. And D. To identify potential side effects of a drug. Very well, guys. Good. So, the correct answer is C. To understand how the body absorbs, distributes, metabolizes, and eliminates. I'm sorry. Sorry. Excuse me. Now, so the correct answer is C. We call this ADME. So now pharmacokinetics, unlike pharmacodynamics, is what once we ingest medicine, how our body starts working with the medicine. Like once we eat the medicine, what about? I think I have have this in the next slide. If I don't, I'll explain every step. Yeah. So this is the next question. I'm sorry, I don't know why that's happening. I hope you guys can see it. Uh, I'm sorry, I don't know what's happening there. So, what does ADME stand for in pharmacokinetics? You guys are pretty quick for this, like, very good. So, I'll just go ahead and explain it. The correct answer is, as you guys state clearly, so it's A, absorption, distribution, metabolism, and excretion. So, yeah. So once we eat the medicine, the first thing that it does is it goes in our mouth. It just basically our body starts absorbing the medicine, absorbing in the different, like, some absorption start right from the mouth. There are few medicines that are easily absorbed, but some medicines they travel around, and the absorption, they have different mechanism of absorption as well. But the first thing that happens is the body absorbs the medicine. Now once the medicine is absorbed, it's in our bloodstream, it goes to the bloodstream, it finally distributes. Now the distribution is via bloodstream. And some of the drugs, I don't know, yeah, let's keep it to that, like distribution. Now distribution, it goes to the bloodstream and it travels through different parts of the body, and it finally reaches its target. And it has to do as well the metabolism part. Now this metabolism part is interesting. uh, once the the medicine, the intended drug, reaches its target, it is metabolized. It's how the it's interacting with the target. And basically all this happens, the metabolism generally takes place in our liver and kidney basically excretion. But what the body does is, like, it just throws the drug in the body. And then it's like a you guys have how, like, take this medicine twice a day, thrice a day, maybe once a day. So that would depend on dosage and the kind of cycle that the drug has. So that's part of metabolism. And finally, once everything is done, the medicine, that's a foreign particle in the body, it has to be excreted. Once its work is done, we don't want that to stay in our body. So it has to be excreted. And now excretion is mainly taken care of by kidney. So that's what ADME stands for, pharmacokinetics. Let's move on to the next question. What does the term half-life, T half, in pharmacokinetics refer to? A. The time required for a drug to be completely metabolized by the liver. B. The time required for the drug concentration in plasma to reduce by half. C. The time taken for a drug to distribute evenly throughout the body. And D. The time required for a drug to reach its maximum plasma concentration. I'll repeat it again. Guys, if you have any question regarding the topic or the question that, you know, if you feel like I haven't been able to explain it properly, please let me know in the chat box, or you can unmute yourself and let me know. I'll try my best to explain it to you. So I think let's move forward. The correct answer is B. The time required for the drug concentration in plasma to reduce by half. So, once our body absorbs it, and it's distributed, once it starts working. So I'm sorry, just a minute. Yep. Okay. Sorry. Yeah. Next question. Which of the following correctly explain Which of the following correctly explains the trend of acidity in hydrohalic acids? Now, HF, HCl, HBr, and HI. I can see a few answers. Well, most of you. So that's good. Most of you got the right answer. It's B. HI, which is greater than HBr, which is greater than HCl, which is greater than HF. Now, let me explain this to you. How that happens. Now, this is due to the decreasing bond strength of HF to HI. As we move from as we move down the periodic table, like in the same group, we can see that the size of the nucleus increases, and the bond it forms with, you know so the size increases here, and as the size increases, its bond with H, it loses its strength. The strength also decreases, and because the strength decreases, it's easier for them to lose that H. And if it loses H easily, it becomes a better acid. So that's why the correct answer is B. HI, which is greater than HBr, which is greater than HCl, which is greater than HF. I hope you guys get it. I'll just move on to the next one. Which of the following compounds is the most basic in aqueous solution? Oh, guys, look through it carefully. The first option is ammonia, second option is NaOH, C is pyridine, and D is aniline. So, yeah, guys, hey, please keep in mind it's mentioning aqueous solutions. So, so the correct answer is NaOH, which is sodium hydroxide. Now, I think So I have explanation for it because I knew this could create some kind of confusion. So you see, sodium hydroxide, it clearly in aqueous medium, it clearly dissociates into Na plus OH minus. It's completely dissociated and hence has very high pH and hence has a strong base. Whereas in ammonia, it's a weak base because it does not completely dissociate in aqueous medium. Instead, what it does is it reacts with water and forms NH4 plus and OH minus. Hence it's not a very good base. Uh, yeah, guys. PPT will probably be shared in my Google Drive. I'll give you guys the link for it once the class is over. If for some reason I cannot check, the NPTEL, concerned authorities will share it with you guys via email or maybe in the forum. Probably my session was the last one. Maybe after my session, they'll send it to you guys. And if they don't do that, I'll probably I'll make a point of sharing everything from my drive once this class is over. Please don't worry about the PPT. I'll share this. Yeah. So I hope you guys don't have any problem with the fact. I'm sorry. Is there any question you want me to repeat? And anything? Please let me know if you're struggling with this question. I can repeat it once again. If not, I'll move forward. Is that okay with everyone? Perfect. Now, I'm sorry, I don't know what's happening. Let me share it once again. Yeah. The next question is, which type of intermolecular interaction is most crucial? Here, the keyword is most crucial for stabilizing the structure of peptide bond within proteins. A. Covalent bond. B. Hydrogen bond. C. Ionic bond. D. Van der Waal interaction. Yeah. Okay, guys. Most crucial in stabilizing the structure of peptide bonds. It's not asking about peptide bond, but what helps stabilizing the structure of peptide bond. Think about alpha helix and beta sheet. That's how the secondary structure probably. I think that would give you a clear idea. The correct answer actually is hydrogen bond. Now let me explain why. See, they're talking about the stability of the peptide bond. Though peptide bond itself is a covalent bond, CONH. Now that's a covalent bond, and that's a primary structure of a protein made up of amino acid. What they're talking about, what the question asked was the stability of the peptide bond, which is the basically the secondary structure of protein, and that is stabilized by hydrogen bond. The CO and NH in alpha helix, they form this kind of hydrogen bond, intermolecular hydrogen bond, that stabilizes the structure of the peptide. So the correct answer is hydrogen bond. Ionic bond, they are very strong and they do not participate. They have um, there are some interaction that are ionic bond, but they are very very strong interaction, and they are not involved in stabilizing the structure of peptides. Neither is van der Waal interaction. They are very weak, and you know at one point it does come into action when you're trying to but keep a molecule in a particular alignment, but it's very weak. So it's basically the hydrogen bond that is responsible for stabilizing the structure of the secondary structure of the peptide. I hope you guys get it. If there's any question, please ask. Now I'll move on to the next question then. I hope you guys get what I'm trying to explain. So now let's discuss the assignment that was from week 0. Um, that assignment was given more or less to give you a basic idea of what are the topics that you might need to revise in order to prepare yourself to understand medicinal chemistry in a better way. So the first question is, you see a molecule, structure of a molecule there. What they're asking is, you have to tell them basically the shape of the marked carbon and nitrogen. What Which set of approximate angles at C1, C2, N of the following molecules indicate correct shape? Now, for solving this question, you guys have to remember the concept of hybridization, overlapping, and everything, and also the concept of resonance. You can write the answer in the chat box. That would give me an idea of, you know, if you guys understand this. So, yeah. So the answer is C1 is 120, C2 is 109.5, and N is 109.5. Now, let me explain why. I'm sorry, just let me see if there's anyone waiting for. No. Okay. This one is H. You can see this one is C1. This one is C2, and this one is N. As you can see, the carboxylic group, it always has a resonance going on its own. So since it's participating in resonance, we can say it's sp2 hybridized, and once it's sp2 hybridized, we know the angle is going to be 120 degrees. What about C2? Now you can see this C does not have any like anything that can like that can extend the resonance coming on from the carboxylic acid. It just has single bond with the 2H, and single bond with N, and single bond with carboxylic C. So here there is there is no resonance, nothing at all. So it's simply sp3 hybridized. Once it's sp3 hybridized, its tetrahedral geometry, it goes 109.5 degrees. What about the nitrogen here? The nitrogen here has a lone pair, but this lone pair is not involved in any kind of resonance, because the C here, the C2, does not have any vacant p orbital to accommodate the lone pairs coming from the nitrogen. So the nitrogen has to keep its lone pair to itself and is hence just sp3 hybridized, and its angle is 109.5 degrees as well. Is this clear to you guys? I'll I see there is no question, so I'll move forward. So which carboxylic acid is more acidic? Have a look at it, guys. Go through the options carefully. See if there is anything that piques your interest, additional thing, and give me the answer in the chat box. Hybridization of that structure. Tanmay, can you be more specific? Are you talking about the previous question? Uh, uh, okay, I can go to. Yeah, which one? Uh, the hybridization. You're talking about this one. So hybridization of the entire compound. So, you know, okay, let me explain it in this way. So when you see carboxylic acid, especially when there is resonance, you know that the lone pair is going to the p orbital, which is vacant. The oxygen, you see, Tanmay, you can unmute yourself and ask me the question more clearly so that I can try to answer in a more productive way. So sp2 hybridization, it's, that means a planar geometry. That's like trigonal, pyramidal, and so once you have planar thing, suppose this is what you want.